entitled Identification of Genes Involved with Blood Pressure Regulation using ENU Technology and Forward Genetics. So, cardiovascular disease is the main cause of death worldwide, and there is, just a second. And there is a relation between, a positive linear relation between BP level, blood pressure level, and cardiovascular risk mortality, here represented by coronary artery disease and stroke. After the studies in Framingham population, and as well as the studies, physiological studies, identifying some systems involved with blood pressure regulation as cardiovascular system, kidney, and neuroendocrine system, it was available a variety of drugs to treat hypertension. However, those drugs treat the symptoms, but not the cause of the disease, treating high blood pressure, but not hypertension. So in clinical practice, we have drugs that act in those systems, but the hypertension management remains challenging due to its complexity that's involved lifestyle and genetic risk factors, as well undiagnosed secondary hypertension, leading to a multifactorial resistance for the drugs, as well as poorly controlled blood pressure in hypertensive patients. As I said, there is a genetic contribution of blood pressure regulation and also hypertension. And usually we estimate the genetic contribution in those phenotypes using family-based studies or twin studies, and even comparing siblings that are adopted and biological siblings. But here I will show you some results that we did here in a Brazilian population by Pendi City that is in Minas Gerais states, where we estimate the heritability of the major risk factors for cardiovascular disease. Here represents systolic blood pressure, so for systolic blood pressure and diastolic blood pressure, we estimated a genetic contribution around 25% after adjustment of age and gender. And for hypertension, we estimate a heritability of 30% here, after adjustment of age and gender also. Remember that those estimations are specific for Brazilian population, because the genetic contribution varies along different populations due to the genetic architecture of those populations. For example, for African American population, the heritability of blood pressure is around 50%. So we know the genetic cause of some types of hypertension in only 5% of the cases. And this type of hypertension is known as monogenic hypertension or hypotension, that usually affects genes, only one gene, mutations in only one gene, and the majority of them affects the kidney system and structure and the self-handling kidney. The remaining 95%, the genetic cause, the genetic cause are unknown, and those cases are known as essential or primary hypertension. That is a multifactorial disease involving environmental and genetic factors, as well as the interaction of both, leading the development of the phenotype. So in the last decades, the scientific community tried to dissect the genetic cause of this complex phenotype. One of the strategies was using experimental models, using linkage analysis, where we analyze a F2 progeny that came from a normotensive, an intercross between a normotensive and a Rex. And we assessed different regions in the chromosomes using molecular marks and the association with increase and decrease of blood pressure. Using this strategy, we identified more than 500 blood pressure quantitative loci associated regions that were associated with increase or decrease of blood pressure. And here I have a figure showing all these regions from rat genome database. And as you can see, there is a genetic or genes involved with the phenotype along all the chromosomes, showing the difficulty to identify the genes involved with this complex phenotype. After the linkage studies, we usually develop congenic strains that are strains developed using backcross of a donor strain that usually is a normotensive strain, but can be a hypertensive one. And we try to transfer only a part of one chromosome in a recipient strain that usually is a normotensive strain. So here are the example of the development of one congenic strain, showing the transfer of this interval in only one chromosome. Using this strategy, we develop a high number of congenic strains. Here is a busy figure, but what I want to show you is that in all chromosomes, each square is a chromosome, and each bar, the blue bar, represents a region transfer from a strain to another, developing a congenic strain. And as you can see, we develop more than 50 congenic strains for blood pressure, and the number of genes that we identify using this strategy as directly involved with BP regulation are very small, as you can see here. So showing that this strategy is not very successful to identify genes and genetic variants associated or related with BP regulation and hypertension. After that, simultaneously as with these studies, in the beginning of 21st century, we finished the human genome sequencing and developed different databases that contain all the genetic variants identifying human populations. Those databases allow us to develop the genome-wide association studies. Those studies try to associate common genetic variants with the phenotype, and this is the first genome-wide association study published in 2007, where they evaluated 14,000 cases of 7 common diseases and 3,000 shared controls, and they tested 500,000 common variants of one kind, that is single nucleotide polymorphisms. And for hypertension, different for the other disease tested in this study, they did not identify any variant associated with the phenotype. So here is the usually presented graph in genome-wide association studies, where we have all the chromosomes tested, and each point is a variant testing that chromosome, and here is the p-value for the association analysis. So as you can see, you cannot see any variant associated with the phenotype. After that, the following studies try to increase the number of variants tested as well as the number of individuals tested. So in this study published in 2011, they tested 2.5 million genotype, single nucleotide polymorphisms. Also, they evaluated 70,000 individuals of European ancestry, and they identified only 29 loci that explain less than 1% of systolic blood pressure variance in this population, that correspond to 5% of estimated heritability in this population. After that, in 2016, They did another genome-wide association analysis, increasing even more the number of individuals used. Here they use almost 350,000 individuals, and they identify 66 loci that explain less than 3.5% of systolic blood pressure variance in this population that corresponds to 14% of the estimated heritability. So the question that remains is, where is the missing heritability? Why we cannot identify all the totality of heritability using those studies? So there are some reasons. One of them is we need a large biomedical data sets considering the polygenic model that those diseases present. So we believe that those diseases are caused by thousands of variants that contribute with a small effect. So we need more bigger data sets to identify those genetic variants. Also, we consider only one kind of method that is an additive genetic effect. So we should consider other analytical methods as non-additive genetic effects, as well as we assess only one type of genetic variants that are single nucleotide polymorphisms. So we should test other types of genetic variants as rare variants or translocations in cells. And also we do not consider in those studies epistatic and epigenetic factors. So the interaction between genes or genetic variants and the interaction of genes and genetic variants with the environment. So after those considerations, the last two studies published in blood pressure using this strategy was in 2018, where they increased the number of people tested to 1 million people and identifying 91 loci that explain almost 6% of systolic blood pressure variance that correspond to 20% of the estimated heritability. And last year, they published this article that used 1.3 million people, and they identify 106 new BP loci, and now they identify also 32 new rare variants. So this is one of the first studies that use genome-wide association studies using rare variants with a very big sample size. However, here is a figure that I can show you some genes identified using this strategy that are involved with blood pressure regulation and hypertension in green, here in cardiac, neuroendocrine, and also in kidney and vascular systems. However, the number of genes identified using this strategy remains low, again showing a very frustrating results to identify genes involved with blood pressure regulation. So, as an alternative to overcome the limitations of those strategies that I presented, we will provide evidence of forward genetic screen strategies using chemical mutagenesis, phenotypic screen, genomic analysis to identify genes that individually influence blood pressure in mice. So for this, we use an ingenious strategy, which the Dr. Beutler lab was pioneer, using forward genetic screening together with next generation sequencing to easily fastly identify the genes that are the mutations that are associated with the phenotype. So here is the idea. His lab generates a pedigree, a family that contain 30 to 100 mice with 30 to 60 handle mutation in coding regions. So we generated a G0 mice that receive the chemical mutagenesis, and the G1 mice present those mutations. These G1 mice backcross cross with his daughters that contain also different presentations of those mutations in heterozygosity. And after that, we obtain a G3 mice that contains a random presentation of all these 3260 coding mutations observed in G1 mice. At the same time that those animals are being developed, the G1 mice have their exome sequencing done, and after that, all the mutations identifying G1 are also sequenced in G1, G2, and G3 mice using IOP and next generation sequencing. And when we start to phenotype those G3 mice, we already have the genetic information. So when we upload the results in the system, we, in some minutes or hours, we have the information of linkage between the genetic variants in those key coding regions and the phenotype. After that, we confirmed the influence of this gene in the phenotype using knockout mice developed by CRISPR-Cas9. So the main goal of this project was to select candidate genes influence blood pressure regulation in mice using a forward genetic screening strategy. And as specific aims, we select candidate genes associated with BP change in pedigrees where 30 to 50 known genes have been mutated. Also, we use reverse genetics to validate each candidate gene using CRISPR-Cas9 strategy, developing knockout mice strains, and also we use the information mouse to search for rare genetic variants that may influence BP in human population with extreme phenotype focusing in resistant hypertensive patients. So the general idea that I want to give you in this presentation is this line of strategy. So first, we use forward genetic screening to identify genes involved with blood pressure. After we confirm the influence of those genes in the phenotype using reverse genetics, and finally, we create a hypothesis how those genes are leading the phenotype, how is the molecular mechanism underpinning BP regulation. So here I will bring the status of the screening process there in Whitewood Lab, showing the results of 20% of the genes in mice genome. Also, we want to show you some validation of the strategy, the forward genetic screen, to identify genes, showing positive controls because we identify genes known to be involved with blood pressure regulation, and also validate some new genes in the involvement with blood pressure regulation using development of knockout mice, and finally showing some hypotheses how these genes are involved with BP. Also, we present a data integration to help us to prioritize the most interesting genes. It's important to say that all genes identifying forward genetic screens are important, and we have biological evidence that they are involved with the phenotype. However, as we have a higher number of genes to study, we use a data integration to prioritize those that seems more important, and also to give us some insights how these genes are involved with the phenotype. Considering that, the idea is that we can redefine hypertension not as a dysfunction of BP control system, but as function of subsystems, as we are identifying genes that are, many genes are in the same system, but in different parts of them. For this, we use the forward genetic screening developed in Whitewood Lab So we use the tech we developed there and also all the infrastructure developed there. And we assessed the BP of the G3 BP using plethysmography, indirect tail curve assessment using this equipment. We have 4 of these equipment in Dallas, and we usually use only 3 of them to assess 18 mice per time. And we leave 1 equipment to rest in 1 week and rotate all of them. The measurement is average. Where is the point? The average of 10 measures for each animal per day, using 5 preview measures as training. We assess systolic blood pressure and heart rate, and usually around using the 3 equipments can take 20 to 30 minutes, and it is possible to do 12 to 26 rounds per day, assessing 200 to 500 mice per day, depending on the person that's there handling the mice. So the week schedule is this one. So on Monday, we mark the animals to make easier to handle them during the experiment. After that, we assess blood pressure in 3 days, and the first day we disregard the data because we observe that day 1 presents a different blood pressure when compared with day 2 and 3 and the average of the last 2 days. Probably because the animals are not used to the equipment and the animal holder. So after that, we assess also on day 2 and 3 and take the average of these 2 days to avoid the daily fluctuation in data. And finally, we analyze data on Friday when we upload the results and analyze the association. So the platform used in Boydler Lab is mutagenetics. Here, we can upload data here. And first, we analyze data considering only 1 family, that is a single pedigree analysis. In this analysis, we identify mutations in code regions in genes in only 1 family associated with the phenotype. So here is an example of the presentation. So we have the gene, coordinates, mutation type, the effect considering polyphen 2, the number of homozygote ref gene. Oh, I'm sorry. The number of animals that are homozygous reference, heterozygous and homozygous variant mice. Remember that here they have this genotype for only this mutation. They have more mutations in coding region. The p-value considering different inheritance, and also the average blood pressure in different genotypes and difference when compared with the wild-type mice for this mutation. When you click the p-value, you can see the Manhattan plot with all mutations and the results of the association analysis, here represented by the p-value of the association and the chromosomes, and each point is a mutation that occurs in this family. And here, as you can see, this mutation reached significant p-value. When you click this point, you can see the raw data from wild-type mice C57 that we always assess together with the families, with the pedigrees, the homozygous reference mice for this mutation, heterozygous and homozygous variant mice. After that, we also consider the super pedigree analysis. There is 2 types of analysis. The first one is the position super pedigree that considers the same mutation that occurs in different families. However, we have a a very low number of cases that we have the same mutation occurring in different families. So usually we consider the gene superpedigree that are different mutations occurring in different families but in the same gene. So the idea is the same of single pedigree. So the presentation here we have the gene, the p-values in different inheritance, and also the average blood pressure of each genotype. And when you place the p-value, you can see the Manhattan plot considering all the families. So here you can see that we have much more mutations, and also much more mutations that consider only one category. And here is the point, the significant mutation. And we can see again the raw data, but here the color represents each family. So the C57 control mice, reference homozygous mice, heterozygous, and homozygous bar mice for mutations in this gene. After that, we seek a candidate analysis first to identify positive controls, so genes that confirm that our strategy is efficient to identify genes involved with blood pressure regulation. And after that, we use prioritization of candidate genes to, again, select those that are most interesting, but all the genes identifying in our genetic screen shows biological evidence to be involved with the phenotype. For this, we use superpedigree analysis, also information from rat congenic strains, and information in human population with distinct phenotype. For rat congenic strains, first we identify those genes that are within DPPTLs in rats. After that, we identify if they are within intervals transferred in congenic strains. And finally, we search for genetic variants in coding regions also in the strains used to develop the congenic strains using a database from Rat Genome Database that contains whole genome sequencing of 27 rat strains. For human population, we use human exome sequencing from target hypertensive patient samples. First, we use data from this sample, the resistant hypertension in African Americans. That is a project available from NIH. And they contain 91 African American resistant hypertensive patients. And those patients were not well characterized because they were selected from a hypertensive sample, and they weren't followed a long time to confirm that they are resistant. We consider as reference population the African American population from GenomeAD, the Genome Aggregation Database, to identify the rare genetic variants in those populations in African Americans. We consider coding genetic variants in coding regions, non-synonymal variants, frame shifts in DELs, variants that are very rare, considering the frequency lower than 0.1% in the African American reference population, and also for missense variants, we consider those that present a Revel score higher or equal 0.5. Revel is a tool that predicts the cause of mutation in our protein. As a new result that I did not present in the printed material, we also finished the sequencing of 450 resistant hypertensive Brazilian patients. Those patients were very well characterized and came from molecular biology laboratory from Rio de Janeiro Federal University, where they followed those patients for many years and try to treat their resistance. For this sample, we compare with a Brazilian database, the Abraão, that contain information from elder people from São Paulo city, and also we consider the data from genome exome, considering all the populations that are present in the database, as our population is mixed, because Abraão is a very small database, and we consider all the same criteria to select the rare genetic variants. After that, to validate the genes, the influence of the genes in BP phenotype, we developed knockout mice using CRISPR-Cas. This is the strategy. So we cause the mutation in C1 mice, develop heterozygous mice in C2, and cross the C2 mice, generating a C3 that had examples of homozygous white type, heterozygous, and homozygous knockout mice in this supergene. And we assess the BP first using the same equipment used in the screening, that is pleximography. And also for one specific strain, we also assess blood pressure using pleximography in low and high salt diet to put another stress in the system. After that, we assess blood pressure also directly, and also we assess bioreceptor function after the basal condition, giving 3 increased doses of a vasoconstrictor, followed by 3 increased doses of a vasodilator to assess bioreceptor function. So the results. We analyze both phenotypes, systolic blood pressure and heart rate. We assessed around 41,000 G3 mice and almost 700 C57 control mice distributed in almost 800 pedigrees, and we estimated that we already investigate 20% of the genes in mice genome. Considering our criteria to select candidates, that is adjusted p-value linear regression using average data, and also data consistent along day 2 and 3, we identify 53 candidate genes for blood pressure and 76 candidate genes for heart rate. Remember here that we have some families or categories that contain more than one gene associated with the phenotype. In those cases, we believe that only one gene is directly involved with the phenotype. So it is why we consider less number of candidate genes that the genes identify. So here I will focus on data from systolic blood pressure. We identify seven genes that are positive controls that are known genes to be involved with blood pressure regulation. Here are the genes, and half of them show increase in blood pressure, and half of them show decrease in blood pressure. And I bring 2 examples. The first one is the mutation in leptin receptor, where the animals show an obese phenotype together with a decrease in blood pressure. This is a phenotype known in human population. There are some human diseases that cause obesity and decrease in blood pressure. And the other example is mutation in MEC8 gene, that animals with mutation in these genes presents cyst formation in kidney, also increase in blood pressure. And again, it is a phenotype observed in some human disease that we have cyst formation, kidney, and hypertension. For the remaining 46 genes, We did the prioritization of candidate genes to identify those that maybe would be the first ones to be investigated. First, we use superpedigree, congenic strains, and also genetic variants between the strains used to develop congenic strains, and rare variants in human population. For human population, we already have here in our lab 4 different populations. The 2 that I already showed you, and 1 other resistant hypertensive Brazilian population, and we have also the UK Biobank database containing 50,000 individuals, in which half of them are hypertensive. But here I will present only results of the first 2 populations. For superpedigree, we have 14 genes that have significant superpedigree, 19 genes that are within congenic strains developed. Only 1 gene that presents genetic variant between the strains used to develop the congenic strain. And for rare variants, for African American population, we identify only 6 genes that present rare genetic variants in those populations, and 18 genes that present rare variants in Brazilian population. The thing, one important thing is the African American population also is a very small sample, and also the sequencing was done in the beginning of next generation methodologies. So the quality of the sequence is not so well. So we believe that the low number of genes with rare genetic variants are due to some of these problems that we identified during the analysis. So here I bring a Venn diagram considering also the rare variants from the Brazilian population. So here we have all the genes that have superpedigree, I'm sorry, the 2A is superpedigree, 2B1 is congenic strain, and 2B2 are genetic variants between the strains used to develop congenic strains. 2C1 is rare variants in African Americans, and 2C2 is rare variants in Brazilian population. So it is a little bit difficult to see, but the idea is that the most informative genes should be here in the middle of the Venn diagram. So we do not have any gene that have all the 4 criteria. However, we have genes that have 3 criteria here that have superpedigree, within congenic strains, and rare variants in Brazilian population, and these ones that have rare variants in both population and also are within congenic strains. Also, we have 14 genes that contain 2 criteria. Those are the genes and the phenotype in green and red. And only to situate you that receive the printed material, those are the genes that have 2 criteria in the other Venn diagram that I show in. So for those, some of them we are developing knockout mice. Here in parentheses, we can see the number of genes that we develop knockout mice. We develop knockout mice for only 1 gene that contains 3 criteria. Here, that is candidate A or tenascin C. We develop knockout mice for 5 genes that have 2 criteria. And we developed knockout mice for 1 gene that presents only 1 criteria, and the other gene did not present any criteria, but showed a very big effect size in the phenotype. So from those, 4 were confirmed using plethysmography, and 4 were confirmed using plethysmography. So now I will focus only in 2 knockout mice because I don't have enough time to talk about all of them. The first one is candidate A, tenascin C. Those animals present no homozygous farm mice present an increase in blood pressure when compared with homozygous ref mice and heterozygous mice. Also, they present, this gene presents also a significant superpredatory. And finally, we observed 5 rare variants in Brazilian population with resistant hypertensive phenotype. This gene is a large extracellular matrix glycoprotein. It is observed that high levels of these genes in areas of wound healing and cancer development and cardiovascular disease. There are some studies showing that this gene is regulated by growth factors and as well as mechanical stress. So these articles show that mechanical stress increased tenascin C expression in fibroblasts, and also tenascin is regulating the stress fiber formation in fibroblasts. And also there are many studies showing that tenascin is upregulated in vascular disease. I bring one example that shows that absence of tenascin C causes acute dissection in mice after stress. So they gave angiotensin II, increase blood pressure, and they develop aneurysm. We developed 2 knockout mice for these genes. The first one presents a 1 base pair deletion in exon 1, and the second one presents 2 BP deletion also in exon 1, and both mutations cause a production of a smaller protein. The first BP assessment using plethysmography did not show any difference of knockout mice compared with the siblings. And here, as you can see, there isn't a difference in weight and systolic blood pressure, neither heart rate. After the BP assessment, after high and low salt diet, we observed a tendency of systolic blood pressure increase in both low and high salt diet, as you can see here, low salt diet, and here. And also, when we did the invasive BP measurement, we also saw a tendency in systolic blood pressure, diastolic blood pressure, and mean blood pressure, as you see here, the average values of each genotype, but without any difference in heart rate. And finally, we show that those animals present a decrease in baroreceptor reflex sensitivity. Here is the graph is inverted, but the idea is that for each change in blood pressure, the change in heart rate for each change in blood pressure. So in both situations, in increased blood pressure or decreased blood pressure, we did not see the same effect in heart rate in homozygous knockout mice when compared with wild-type mice. Our working hypothesis is that tenascin affects vessel wall remodeling, altering the extracellular matrix, causing effect in baroreceptor sensing capacity. As you can see here, the baroreceptor sensing is in the aortic arc. So maybe with the remodeling of the vessel wall in this region can cause the effects that we are observing in the baroreceptor reflex, leading also to an increase in blood pressure variability. In summary, we did not observe any difference in systolic blood pressure in knockout mice for tenascin in control conditions, but we observe a tendency in systolic blood pressure increase in knockout mice when compared with wild type in low and high salt diets. Also, we observe the same tendency in using direct BP measurements after a salt intake. And there is a deficit in baroreceptor function in tenacin knockout mice. As next steps, we will assess the efferent, central, and efferent components of the baroreceptor reflex. Also, we want to assess the structure and function changes in aorta, assessing collagen, elastin, vessel reactivity. We want to assess mesentery arteries, heart, and kidney to search for evidence that tenacin C leads alter mechanical properties that are consistent with the change in baroreceptor function. The second knockout is candidate 31, ADO. Knockout mice, the initial screening showed that homozygous VAR mice for mutation lesions present a decrease in blood pressure when compared with wild type and heterozygous mice, as you can see here. And this difference is around 30 millimeters of mercury. This gene codifies a cysteamine dioxygenase enzyme, which plays an important role in homeostatic regulation of cysteine levels and provision of hypotaurine and that oxidizing taurine. There is another protein involved in this pathway that is cysteamine dioxygenase, but it is observed that cysteamine dioxygenase is one of the most important proteins to produce taurine in heart and brain. Taurine is a non-essential amino acid, and there is evidence that when you give taurine to spontaneous hypertensive rats, as well as for hypertensive patients, we have a decrease in blood pressure. So there are some evidence the involvement of taurine in the blood pressure regulation that is involved with nervous system regulation, leading to a decrease in mean arterial pressure and bradycardia. Also, they regulate catecholamine, and they are the taurine production is regulated by baroreceptor reflex, and also there are evidence that taurine regulate plasma vasopressin levels and production in the brain. So we developed one knockout mouse for this gene that has one lipid insertion in exon 1, leading a patient that produces a very small protein. Those animals present a decrease in blood pressure, followed by an increase in heart rate. And we think that this is a counterbalanced response to the decrease of blood pressure. So here you can see that the blood pressure is very low compared with wild type and heterozygous mice, and the heart rate is very high without difference in weight. However, we expect an opposite effect, because if you don't have cysteamine dioxygenase, we expect to have decrease in taurine production. However, those phenotypes seem similar when you give more taurine for hypertensive patients and animals. So we imagine that maybe the cysteamine dioxygenase is upregulated and leading more production of taurine. So our working hypothesis is that hypotaurine is elevated as well as taurine, leading to a decrease in mean arterial pressure, but without bradycardia. We are observing a tachycardia as response to low BP, and we expect that we have hyperactivation of sympathetic system, trying to compensate this low blood pressure. In summary, we have evidence that knockout mice show decrease in blood pressure followed by increase in heart rate without difference in body weight. And as next steps, we want to assess baroreceptor reflex sensitivity, vasopressin modulation, assess afferent, efferent, and central components of baroreceptor reflex, expecting to see an increase in sympathetic activity, and another set points of baroreceptor reflex, assess hypotaurine production and expression of dioxygenase in central nervous systems to check if we have upregulation of the other protein. And also, we want to assess a knockout mice response to beta blockers. So we expected a higher decrease in blood pressure in knockout mice when compared with wild type, and high salt diet, observing also a decrease in sympathetic activity and increase in blood pressure. So in summary, we here, using this strategy, we show biological evidence for genes influence BP in mice through a systematic analysis of the genome. We speculate that we are identifying core and hub genes, since individually those genes have a big effect size in blood pressure phenotype. Also, we present a unique opportunity to understand novel mechanisms whereby those genes lead to BP phenotype, and we speculate that maybe using this strategy, we may give rise to redefinition of hypertension that is known as a dysfunction of BP control system to a dysfunction of subsystem. I'd like to thank Vigor's team, everybody here from the lab, especially those in red that are directly involved with hypertension and blood pressure projects. Also, I'd like to thank Berkeley Lab team that helped a lot and developed all the mice that we use in this project. And also I want to thank the animal facility from University of São Paulo Medical School that are taking care of all knockout mice that I brought from the United States to Brazil, and to my family and friends. Thank you.
Thank you, Samantha. While you remove this from the screen, take your seat here. You have a glass of water. We're going to prepare for the second part of this event. I just want to remember that there are people following us from, they are, I believe, to not Skype, to the YouTube. So I will make sure that I will introduce our friends. So we have a panel of distinguished professors that will contribute to this event. We're going to start the discussion with Professor Bruce Beutler. Professor Beutler is from UT University of Texas Southwestern Medical Center. He's the director of the Center for the Genetics of Host Defense, and also he holds a chair in cancer research. He has a variety of interests, but one of his main interests is in immunology, and he's have important contributions, and some of them were well recognized in 2011 when he received the Nobel Prize in Physiology for Medicine. So, Bruce, please, you may start your argument with Samantha, and you may choose to do on a dialogue or a series of questions followed by answers.
Well, thank you very much, Jose, and thank you, Samantha. That was a lovely presentation and a very interesting one. And I have to say it was a pleasure hosting you in our laboratory. You worked very hard and you did a lot of very interesting stuff.
Thank you.
I want to start with a sort of general question that has no answer, but hear your thoughts about it. For many years, people spoke of the common disease, common variant hypothesis, the idea that genetic diseases that were common, like hypertension, were caused by common variants. And you have discussed a bit the different rates of success in identifying causative mutations in rare versus common genes. What's your conclusion overall? Is most hypertension caused by rare variants or by common ones?
Well, I think in human populations is a little bit different from mice phenotype, but in human, I think it's the combination of both things. We have thousands of variants contributing very small that are common and contribute with a small effect size, but also we should have some rare variants that contribute with a big effect size, but as they are rare, it is difficult to identify using the strategies that we are using now. And also we need a bigger sample size to identify them. And maybe they are involved with those cases that we have extreme phenotypes. So it is why one of based on that, it is one of the ideas to study the hypertensive patients. So in those individuals, maybe they accumulate rare variants with big effect size. But for human population, I think it is more a combination of both things, not only one or only common and rare. I think this is it. And also because assessing the common variants, we are not reaching the totality of heritability. So we are losing something that we are not able to detect. And in mice, we can identify those genes that have a big phenotype. Maybe they are involved with the disease in human. But one thing that we can use those data is that they can identify gene networks or subsystems that are affected in human population, not exactly with rare variants, maybe a combination of both things, but with this strategy we can how can I say that? Highlight those pathways. I don't know if I answered your question.
It comes down to the answer that I interpret is that there's a mixture of rare variants, which may be more extreme, and common variants, which may be less so. And it's very hard to tell what the relative proportion is at present. Is that a good summary?
Yes.
Well, if we think about the common variants and about diseases caused by common variants, those variants got to be common despite the fact that they cause a disease. And that implies that they have some kind of beneficial effect as well. And what would you say might be the beneficial effect of mutations that are common and that cause hypertension?
Beneficial? Well, maybe not today, but if you have hypertension when we breed in the forest, maybe they show some good phenotypes because you can handle, I don't know, but I don't know if you can see any beneficial effect. I don't see like this. I think that probably those mutations, they are not beneficial, but they pass as you cannot notice by natural selection because they do not have so much change, but as today we are living more, and we feel the response of those genetic variants. So I don't know if there is any beneficial thing now in our society. I can see only bad, negative response, but maybe I cannot see. Maybe in the past.
It calls for some guessing, of course. And your point about how it's a different world for us now as human beings than it was in the wild state even a few thousand years ago, that's absolutely right. But perhaps some of them contribute to the fight or flight response. In fact, we know they do, right?
Yes.
Some of them probably make us more fit in one way or another, and nonetheless, you sacrifice many years of life to attain the ability to survive and reproduce at a young age. I think the question calls for a lot of speculation. Is there anything else you want to add to it?
No, I think it's exactly this. Maybe it is a change of our living world now.
Okay, all right. I'm sorry you can't see me. I'm smiling because I'm asking you somewhat impossible questions to answer. Would you say that in general, looking at all your mutations, that you saw more that were purely recessive or more that were semi-dominant or dominant when you were going through all the ENU-induced mutations? Did you usually have a heterozygote effect or usually not?
No, usually I saw recessive phenotype, but I have some examples. The examples that I gave in the thesis from quarter 1A, the inheritance was dominant, and also I didn't show all the mutations that we generated until now. I put a threshold in 20% of the saturation, but we do identify other mutation in quarter 1B, and also the phenotype was presented as dominant. But usually we saw the phenotype being recessive.
Were there any mutations that you could say categorically this is a gain of function mutation?
Gain of function.
Of course, these are quite a lot more rare than loss of function, but they do happen.
Yes.
And usually they're dominant. But it takes some analysis to be sure that you've really made a gain of function allele and you've increased the activity of the protein that was modified. Do you have such an example?
An example. I'm trying to remember. There are so many genes. I can't remember.
I was thinking about it in connection with your ADO mutation, because there is a bit of a paradox that you would expect that giving taurine should lower blood pressure. And here you have a case where you knock out a gene that is supposed to be required for making taurine in target organs like the heart. And yet you have this strange effect that you have a lowering of blood pressure that way too. And I didn't quite understand your explanation for it.
Yeah, I don't know if it is a gain of function, because when the knockout develop contain a stop mutation at the beginning of the protein.
So we could assume that's not going to be gain of function, the knockout.
Yes, I don't think it's a gain of function. I believe those animals present absence of ADO. But as I showed in the pathway of production of hypotaurin, we have two different proteins that are involved with hypotaurin production. The cysteamine dioxygenase and the cysteamine dioxygenase. So maybe the absence of one protein caused the increase of production or expression of the other protein. I don't know. It is the only idea that I had when I saw those results, because I did not expect the decrease in blood pressure in those mice.
Did you try to measure the taurine in blood or in tissues?
No. Those animals, we are bringing the sperm of those animals now. So I'm waiting to receive the sperm to revitalize the mice here, to check this, because we need to check if they have increase of taurine in plasma, and also if they have increase of hypotaurin in the nervous system and heart.
Okay.
But, yeah.
If I got it right, you had 53 mutations that change systolic blood pressure that you have declared. And what fraction of those increased blood pressure and what fraction decreased blood pressure?
I think the majority of them, I think it's half-half, almost half-half. Half decrease and half increase. What I noticed is that when you have a decrease, this is for heart rate, not for blood pressure, because when we assess heart rate, usually when you see the phenotype, the mutation that causes decrease in heart rate, we have a very huge phenotype, but for increase in heart rate, we do not observe so many different, not so big effect size as decrease.
Yes, I noticed that myself, and I wondered what your explanation for that might be.
Well, I think it's more difficult to increase heart rate than decrease heart rate. I don't know.
The stress of the phenotype.
Oh, yes, because our the experimental conditions that we use are
and there is a bias.
We stress the animals with all the system with the animal holder, so I think we increase the stress.
Then your assay is more sensitive to picking up the bradycardia mutations.
Yeah, I think so.
Yeah, that's a fair answer. That's good.
Yeah, and maybe it is the same for blood pressure, because we are more sensitive to see difference in decrease in blood pressure than in increase in blood pressure, because the stress condition also increase blood pressure.
My next question has to do with how you classified things as known causes. You said that out of those 53, there were 7 known causes of altered blood pressure, and you listed them. And I looked at your list, and I saw that it did not contain an example that you had shown earlier in your talk, which was urate oxidase deficiency. You showed the example of urate oxidase.
Oh, yeah.
And I would have said that must be a known one. I would call it, well, that's obvious. You get a urate nephropathy, and then you get hypertension. But you didn't count it as known. And why was that?
Yeah, it is difficult to consider what is positive, because when I consider positive is when we have a lot of events, but sometimes I don't know exactly if I can consider as positive or not. So we have other examples also. I think the one receptor that there is some evidence that this gene is involved with blood pressure regulation, but when they developed knockout, they did not identify any phenotype. So I don't know if I consider as a positive or not. but I think I sometimes I missed some candidates.
Now I will ask you some difficult questions on which I am a particular expert. And let's see how well you know how to calculate saturation in these ways. And you talked about how we have reached 20% saturation, and I want to know how you know that and what you mean by it exactly. In other words, are we talking about only loss of function mutations or loss of function and gain of function both? And when we say saturation, what are your standards? How many times did you see homozygotes with true loss of function? Go ahead, discuss saturation with me.
Okay, I have difficulty with this information. When I was in your lab, I tried to talk with many people, and I don't know exactly how we calculate that, but this number that I show you in presentation is considering not only probably new mutation, but probably damaging mutation. So I think maybe we are considering some gain of function also, but I think. And also I select only mutations that occurs in at least 3, in homozygosity in at least 3 mice. But I don't know exactly how to calculate that, and I took this number in mutakinetics.
Well, that's a very good number if you chose n equals 3. That's pretty stringent. And the way we assess saturation is using a standard where we take a curated set of genes, about 4,000 of them, where it's known that robust knockouts, so loss of function, is lethal in the homozygous state. And we looked at many, many thousands of ENU mutations that landed in that set of genes and were separated by at least 100 megs from any other mutation we identified. So out of 250,000 or so mutations, we had a few thousand such instances. And then we would ask, did this mutation cause at least a significant loss of the number of mice born with homozygosity at weaning age? And in that way, we were able to judge for thousands of mutations whether they truly caused significant damage, enough to affect a complex physiologic process, survival to weaning age. And we take those mutations as representative of others. And we can assess using classical criteria whether they're likely to be damaging to the protein or not. And it was using those scales that we got true percentage likelihood of damage to a significant degree, mutation to phenovariance. That's something I think about every day, and you don't think about it every day, so I'm more able to say it. But
yes.
What you did was use a good criterion for lethality, and you figure then that you're at 20.02% saturation, and you have 53 mutations that affect blood pressure. What is your conclusion? Are there almost exactly 250 mutations or 250 genes in all where mutations can cause a phenotype?
Yes, is what we think. That probably we have around 250 genes that when you cause a point mutation, that a loss or gain of function, you can identify a huge effect size. Maybe this represents some subsystems that are affected in hypertensive disease. I don't know, but it is pretty much what we think. I don't know also, we are using a simple mathematics, but we know that we are increasing the number of animals that we are developing and testing, and when we reach the 80% of the genome, I think it would be more difficult to identify genes that affect the phenotype. Because when you don't have any evidence, you have many points to focus. But when you have many points that you already identified, I think it's difficult to identify another one. But I think it's probably 200 genes that are involved with the phenotype.
Are you at a point of combining them, combining mutations to see if you get synergistic or at least additive effects?
You think using knockout mice?
Yes, I think you should reach a point of understanding where you can say, let's say this gene is involved in controlling heart rate, and this one over here is involved in controlling vascular tone. And if I combine the two of them, probably I get a super additive effect.
It is a very good, interesting point, but I don't, maybe when we have more information of the same pathway, we can analyze things together. But my afraid is that when I combine two mutations, mutations that cause a big effect, if I can develop variables mice, I don't know, maybe it is difficult. I don't know, are you trying this in your screenings? Because it is possible to do that.
Yes, certainly sometimes we do try to combine them. If we have a hypothesis about how they work in general, we try to test that hypothesis. And one can make certain predictions. Of course, we're not always right, and I expect you would not always find additivity where you expect it, but at some point when you really have high confidence about a large number of genes, but not a good mechanistic understanding, it does help to try to combine two or even sometimes three mutations.
Yeah, I think it's an interesting experiment, and maybe to elucidate even more some pathways and systems.
All right. Well, thank you for those answers, and I will yield to the next questioner.
Okay, thank you, Bruce.
Thank you, Bruce.
Yeah.
Dr. Barth.
Just one comment. It's very hard to be sitting here and not talking, but, because the discussion is very good. But one thing that even with the 20% of coverage in these 46 genes, your last questions regarding combination, we are not able yet, even though we are doing a lot of integration over here, to try to identify the subsystems that would allow us to have a hypothesis to combine genes. But we just finished the sequencing of the 450 individuals with resistant hypertensives, and 170 more should be completed, at least the sequencing, until the end of this month. So, assuming that we have combinations, consistent combinations of these candidates that are appearing there, I would then, even not knowing precisely the molecular underpinning, be tempted to combine them on a knockout mice, let's say 2 or 3, based on what we see in terms of clustering of rare gene variants in these patients.
Okay. Yeah. I think at some point you've got to, and it would be really nice to see where you're right and where you're wrong and whatever ideas you have about how the systems operate together.
Exactly. Well, thank you so much. So now we're going to pass to Marcelo. Professor Marcelo Nobrega is a professor of human genetics at the University of Chicago. And Marcelo began as a physiologist. And when he did his postdoc with Ed Rubin, he became interested in genetic architecture, defining complex phenotypes. And one of the things that he first tested there, successful, was to try to answer the question of that at the time, in the 90s, we would call desert areas of the genome. And he started producing transgenic mice to test what are the regulatory sequences contained on those genes could interfere with specific phenotypes. From there, he began a very profitable career at the University of Chicago in the human genetics department, and he has followed through on that, and his main interest now is trying to dissect complex architecture of genetic architecture of complex phenotypes. And he has some interesting data recently, especially on obesity and teasing out this mechanism. So thank you so much for your participation, Marcelo, and you two may start the conversation.
Okay, thank you very much, Zach, and Samantha, congratulations on your great presentation and on the thesis as well. It's very well written, it's very linear. You did a good job, and it's always a pleasure to get a polished document like that to read. So, Samantha, I wrote down a few questions in the order in which they were appearing in my mind as I was reading your thesis, and now I see that some of them partially overlap with the questions that Bruce already asked, but I'm gonna try to give a different angle when possible to those. Okay, so in the beginning of your thesis, in the introduction, you talk about how difficult it was to ascertain what genes were involved in a trait, like such as blood pressure, because the designs of the human studies, such as GWAS, were disappointing in some way, in that after we put together all the information that we're getting from the GWAS for blood pressure, we're still left with just a fraction of the heritability accounted for. And so my question to you is, like, can you talk a little bit about, and you actually do that in the discussion, I have to say. So what are the factors that reduce our ability to account for heritability of blood pressures in GWAS studies? Where do you think is this missing heritability?
Well, I present some in the presentation. I think one of the cases are the genetic, the type of the variant that we are using. Now we are trying to change a little bit, try to identify rare variants involved, but usually we test single nucleotide variants, or now we have some effort to use insertions and deletions, but I think maybe we have other types of variants that are involved with those diseases and we are now testing. I think also we are improving the number of the sample size of our analysis to increase the power of those analyses. But one thing that we not consider that I think is important is the relation between genetic variants and genes, and also epigenetics. I know that you work a lot with those things, but I think we should try to improve our methods to consider also the influence of the environment and also the relation and the interaction between variants to lead the phenotype. So those things I think are very important. And together with the not testing rare genetic variants that I think maybe for some cases are the case and are the cause of hypertension development, maybe those three things I think would be important to understand the totality of heritability of these complex phenotypes. And I think we are doing a lot of effort to try to integrate data considering some epigenomics marks as well as EQTLs to try to identify how those genes interact with the environment that we are. But maybe we need to improve even more to understand what does mean.
Okay, so also like, and you touched upon this, right? I mean, one of the critical things that has happened is that our ability to do these really large studies, these meta-analysis of many studies for each one of these diseases, and in the cases of those, like for example, for obesity, where you try to do polygenic risk scores for obesity, and you get like the people that have the top decile of the polygenic risk score. You see that 85% of these people are obese, not just like a little heavier, but significantly obese. And in the accountability of the heritability in these cases approach already 60%. So, I think that what's important to know is that maybe unfortunately, it's not that these missing heritabilities elsewhere or in the form really of rare variants. It is in a really large network of variants with the small effects that eventually, if we keep doing these ever bigger studies, you're going to go from 900 loci associated with the blood pressure to thousands. And the question is, how many of those matter, at the end of the day?
Can I include a comment?
Please.
We are working with polygenic for hypertension, for blood pressure, for systolic blood pressure also. So there is a master student here that is developing the polygenic risk score for blood pressure, and we are using much more genetic variants that were significantly identified in GWAS studies. We are using almost 500,000 variants, this polygenic risk score, using the same strategy used in that paper for obesity. And what we identified that using those almost 500,000 variants, we can explain 10% of blood pressure heritability. So we double the capacity to explain the phenotype. So we identify almost 50%, 40% of heritability of blood pressure heritability, but it's still missing something that maybe is not on common variants that explain very small part of the phenotype, has small effects on the phenotype. So we are trying also to assess that here in the lab, and it has been very interesting to work with all these ideas that maybe we are not considering all the common variants involved with the phenotype when we consider only the significant one in genome-wide association studies.
Okay, great. So let's move on. And I just wanted to say also up front that some of these questions will be a little bit provocative, but what we're trying to provoke here is just your thoughts, right? I just want to assess how you're thinking about the stuff. It's not that I'm doubting at all what you did, okay? So in the thesis, you summarize the effort to map genes associated with blood pressure, both in humans with GWAS or in animal models with QTL mapping or congenic strains, with a statement. I'm going to read from the statement. It says, many genes with small effect sizes on the phenotype and high interaction between them are underlying these traits, probably. And therefore, what is lacking is a strategy that directly identify new genes which have a large influence on blood pressure and consequently on hypertension development, which sounds very reasonable, right? And I think it's a very smart strategy. Let's go directly to the genes that have a big effect, because those are the ones that are likely to inform us about the processes and maybe become therapeutic targets. And that would, in principle, seem to exclude these small effect variants from GWAS. But let's consider one example from GWAS, which I think is very informative and not just like an exception. I think this is actually the rule and represents one of the challenges of using GWAS to understand the architecture of human diseases. If you look for the GWAS for blood cholesterol levels in humans, okay, one of the strongest associations is a variant in the promoter of the gene that encodes for HMG-CoA reductase. Right, the protein that comes from this gene is, of course, the target of the statins, right, the blockbuster drugs that dramatically lower cholesterol levels in people. However, if you take a group of people that have the high cholesterol alleles and compare with a group of people that have the low cholesterol alleles, okay, and you ask, what is the difference in cholesterol levels between these people? And the answer is that less than 2 milligrams per deciliter. So it's a minuscule effect, right, that would have absolutely no importance in the clinic or predictive value by itself at all. And yet, this is the locus that is highlighting HMG-CoA reductase, which is the target of a blockbuster drug. So here's the question finally. How do you reconcile this apparently discordant evidence that we see in GWAS of small and large effects of these locus on cholesterol levels?
I don't know if I understand well the question. Can you make again?
Yeah. Why, when you look at the GWAS data, you see that the effect of the SNP that is associated with cholesterol levels is so small. It changed cholesterol levels by less than 2 milligrams per deciliter. And yet the gene has the ability to cause a very dramatic change in cholesterol levels if you were to treat the person with the statins. So in principle, looking at the GWAS, you would never guess that this gene is a viable target for therapeutic development, but it is.
Well, I think it is the same thing of hypertension. There are some, because it is a gene involved with cholesterol production and regulation. So we know that this pathway is involved with the development of the disease, and we don't know exactly how the genetic causes are leading the difference in this pathway or system. But we have drugs that act in those pathways, but they not act directly in the cause of the disease. It is the same for hypertension. I think we have good drugs that act in systems known to be involved with blood pressure, but they don't explain the totality of heritability, because you have a regulation of different pathways and systems. So I don't think the small effect size of a variant in a phenotype can say something about the drugs available that acts in that pathway. But the idea, I think it's the opposite. Don't consider those drugs, but obtain new evidence that we can develop drugs that act precisely in the cause of the disease. I don't know if I answered your question.
You do. I mean, you did. But I think that the kind of cautionary tale that I would bring up is the following: is the understanding of the usefulness of GWAS with small effects. Why is my laboratory chasing these things, for example? And it is to remember that what GWAS is telling us is that these fortuitous variants, almost all of them in regulatory elements, right, in regulatory DNA, cause ever so slightly changes in the expression of a gene, in the regulation of a gene, such that at a population level, you can already detect this difference. But this tells nothing of the ability of that gene in the protein, obviously, that is encoded by it, to regulate the trait. So when you give a statin, you are amplifying the effect of basically blocking that gene to 100%, and then you get a big effect, right? Whereas the GWAS, the naturally occurring common variant, is only changing maybe the expression by a couple percent. So the challenge really is of all these GWAS, in my view, from all these GWAS loci, like the 900 associated with blood pressure, to infer which one of those are connected functionally to genes that, if we were to manipulate, would cause a big effect. And I think that this is a very interesting point, since you went to the gene. So in my view, all the work that we do to identify causal variants and finally link somehow these variants to the expression of a gene, all that part is useless. I mean, it's only when you have information about the target gene and where the change in expression of that target gene is associated with disease that you really start doing the biology. And I think that's where, in a very smart way, you're starting from that level because you're already manipulating the gene. So what I wanted to say is that I think that a giving emphasis on the GWAS associations is as good a strategy probably right now for you as focusing on the rare variants, because you are quite underpowered to look at the rare variants with the numbers that you have, right? You're familiar with the work from the giant consortium that sequenced 185,000 genomes of people that had diabetes, and find that by and large, it was not rare variants that were accounting for the heritability. It was the common variants with small effects. And they weren't able either to increase very much the number of genes that were coming out from the rare variants compared with what was already known from the association. So you may have already a very good catalog of candidate genes to focus in your hands with the GWAS, even before, and I'll come back to the questions that I have about the rare variant ideas. So here's one question that is also provocative. So if you have about 900 candidates already from GWAS, and after these 40-odd thousand mice that you had to generate a phenotype, which sounds like I cannot even imagine like the work behind getting all this done. Would it be feasible that a straight up CRISPR approach where you took out these genes would be more cost effective and reduce the phenotyping needs if after 40,000 mice, you still are down to 20% of the loci? So have you thought about these? Now that making these CRISPR knockout mice have become trivial, why not just focus on the genes that are candidates?
Yes, we should. Yeah, you can focus only on these 53 genes, and we have much information about pathways that are involved with blood pressure and confirm them. I think some of them we cannot confirm because we cannot use knockout to understand how they are leading the phenotype, because we take them off in the beginning of the phenotype. But one thing that we want to know, to understand, is what's happening the hypertension phenotype. Because I don't think if you focus only on those genes, I don't know if I understand correctly the question. If I am not responding you, please let me know. But I understand that you are questioning that why we not focus only on those candidates.
No,
right?
no. If I may, the question is the following. Why instead of going to Dallas and doing 40,000 mice to be able to get 53 candidates, you didn't order just knockout mice?
For all the...
For 20% of the genome. Would you have the same result or not? Let's say that money was not an issue.
Okay.
And Bruce said, come to my lab and do this, or you say, go to a company and order 20% of the genes. Would you get the same result or not?
No, I don't think so.
Yeah, so that's
what
he wants to know.
Sorry.
Yeah, that's what he wants to know.
So no, I don't think so, because the beauty in the system developed in the laboratory lab is first the randomizations and probably the mutations that are caused by ENU are not the same as a knockout mice. Sometimes you can cause a loss of function that I think is maybe the majority of the time, but sometimes you can cause the gain of function. So I think using a knockout, you aggregate the gene, the beginning of the development of that mice, and sometimes you have ways to compensate the absence of that gene. So I think the other strategy maybe bring the possibility to analyze alterations that are not so blunt as knockout. So I think we won't obtain the same results. And one evidence of that is that we have some knockouts that does not confirm the phenotype. And we don't know if it is because they actually are not involved with the phenotype, even though we saw the biological evidence in ENU, but or if it is the method using knockout that not allow us to see the phenotype, because you take off the gene at the beginning of development of the mice, because you have other genes that compensate the absence. So I think, no.
Yeah, I think these are very useful, very, very palatable explanations, Samantha. With the ENU, the mutation is also germline, right? I mean, you're looking at you're ultimately phenotyping the G3, which got the mutations germline. So that's not different than a CRISPR, a straight up CRISPR mice. I think that the nuance of a single nucleotide change that may be either generate a hypomorph or a full loss of function allele, or in the maybe more interesting cases, a gain of function is a difference, a significant difference with the knockouts. However, you are following up those observations by making knockouts. So that's something to be discussed also.
Yeah.
But let's.
It's a limitation. It is a limitation of process. Maybe in the future you can try other methodologies or the overexpression of a gene. I don't know.
I think that ultimately the justification that you're giving is good, but it has to be framed on the motivations for your study. So I can see a few of possible scenarios for motivation. One, which I think is what you are interested in, correct me if I'm wrong, is to understand the functional architecture of high blood pressure or blood pressure regulation, right? So what genes are involved in this phenotype? Another one would be to what genes are involved in high blood pressure in humans, where genetic variation causes high blood pressure in humans. that's the one that if that was your question, you might give a second thought about making the GWAS a stronger criteria for going after, right? And a third one, and we're going to come to this in the next question, is are you looking for genes that might become druggable targets? So, and that gives rise to the next question. So, you know, why your choice of genes to follow up from your screen with knockout mice? Use comprehensive annotations like GWAS, rare variants, and other criteria. One of the criteria that you could have thought about using would be the likely druggability of the target of the gene. So genes encoding for proteins that are membrane receptors, ion channels, enzymes with catalytic pockets, right? So basically the targets for most of the therapies that are out there for blood pressure, for high blood pressure and many other diseases. But some of the genes that you chose to follow, and when I wrote the question, ARID4A came to my mind because that's what it had already emerged at that point. These genes are broadly expressed and have global impact on gene regulation. So ultimately, ARID4A, all these ARID genes are involved in chromatin state, and will have a large impact on functions, on gene expression and global gene expression in the cell. So antagonize or inhibiting a protein like that, like ARID4A, will likely result in many unintended consequences besides lowering blood pressure. So this is something that we struggle in the lab all the time, which is defining what are the next targets that we're going to go for. So how do you perceive the selection of new targets for investigation going forward? So now you've done all these and you've got already a bunch of targets, but you're going to presumably continue, or the lab will continue to screen for more genes. So and it's the function follow-up of these that will take a lot of time. So how do you plan to pursue these in the future? What are you going to use the same criteria? You're going to adjust for possible therapeutic targets? How are you going to do it?
Well, the criteria, I think the criteria are changeable. We change a little bit, even since the beginning of the project, and we are considering other informations also. We are working on other informations here in the lab. So one of the things is we think that the GWAS studies can bring a very interesting idea of dysregulated pathways. So one of our efforts is try to understand how those genes identifying mice and in GWAS studies are talking with each other, doing some gene network analysis, considering GPEX data with expression data from different tissues in human. Also, we are analyzing the mutations that we are observing in the mice to see if you can see any pattern in the protein, and doing some computational analysis also to identify any evidence that those mutations causing some effects in function. So there are some ideas how to improve our criteria, and even the rare variants that we are identifying in human population, we are trying to test a hypothesis. So we starting searching for rare variants exactly in the same genes that we identify in mice, but the idea is expand that for genes that in 2 or 3 levels that are interacting with those genes, and also do some burden analysis, have some statistics that those variants are hyper-represented in the individuals with resistant hypertensive, and it's not only a random finding. But I think the criteria that I use here in the thesis is changing. We are trying to integrate all the information that we can in our analysis to see, to prioritize the genes or even understand the pathways or the subsystems that are dysregulated in this phenotype. So we are trying different strategies, but all the strategies focus in only one thing. And one of the ideas that we have is create a platform that we integrate all the data from human for computational analysis and also from mice obtained in Beutler lab in the same place to see which genes have more information. And regarding the drugs, we have also those information, but I never paid much attention on that, and maybe you should pay more attention on it.
Yeah, well, it depends. It depends on what your, like I said, your motivation is. In the understanding the mechanism by which blood pressure is regulated is a perfectly defensible motivation. You don't need to be curing hypertension, so it's just food for thought. Yeah. So let's talk about these. Let's talk about these rare variants again. So you looked at these rare variants in a cohort of resistant hypertension, right? In patients with resistant hypertension, and you found candidate damaging variants in 7 or 8 genes from your cohort of mice. But when I was looking at these, I saw that some of these mutations in mice, and I wrote down here 2 genes, CFTR and CORO1A, that the mutations in mice led to reduced blood pressure, which seems to be like if the candidate mutation in humans was in those genes was causative of the high blood pressure, you're now seeing an effect in the opposite direction. So how do you reconcile this finding with the challenge of inferring whether the rare mutations in humans, where your only power right now at best to identify one individual harboring a rare variant per gene, right? How do you know that these are going to serve as a useful parameter? You talk about mutational burden. So how do you plan to do this to cross-validate your findings and define a set of candidate genes to follow up, with the danger of like missing the market, going after a gene that is really a fortuitous association with humans? So how are you going to do this from now on once you get these few hundred extra genomes that you guys are currently analyzing?
Well, this is something that, of course, we have some genes that present low blood pressure. The mutation leads to a decrease in blood pressure, and we are finding patient in hypertensive patients that probably we hypothesize that they cause increase in blood pressure, but again are not the same mutation, so we can cause a different phenotype depending of the mutation that we are observed in mice and in humans. So I don't think this is a big problem, but the problem that we are facing studying the resistant hypertensive, it is challenging because we are identifying some genes that have mutations, but usually we have 1, 2, or 3 patients with the mutations. I don't know, one of the idea was use burden analysis to bring a statistical analysis to see if what we are identifying is significant, but I don't know exactly what we will get with that. We are learning how to do this analysis now, and I didn't finish yet. But I expect that with the increasing number of the individuals that the resistant hypertensive patients that I analyze, maybe I can have more power to infer some evidence that the contribution of these rare variants in human involving the alteration function of those proteins and genes and leading to the phenotype.
Okay, very good. So the next question you probably already answered right now, just now, and it has to do with one of your candidates, which is the candidate 8. So it's a gene that a gene that a null mutation. I think that you said in your thesis that the mutation gives rise to a truncated mRNA, and that there's good reason to think that this is a null mutation, and this led to increased blood pressure. But when you did your knockout, you didn't see any blood pressure phenotype. I mean, any blood pressure, direct blood pressure phenotypes. You saw the altered barrier reflex changes. So, are you comfortable with the thought that this is probably due to the different nature of the mutations?
Yes, I think there are two things. First, it is the nature of the mutation. I think both mutations are different. The mutation in EMU mice, they cause stop codon, not in the middle, but in the 3/4 of the protein. And the knockout, we do not have the protein. We have a very small part of the protein. So in those cases, again, I think that the absence of the protein can be somehow, how can I say it?
Explaining the differences in phenotype.
No, when you cause the knockout, you don't have the protein. So sometimes
the other one is a hypomorph.
Appear and do part of the work that that protein.
Hypomorph, yeah.
Yeah, yes. So I think this is the case. Also, in EMU mice have other mutations, of course, that are not the same mutations in all other mice, but we have other mutations that can contribute to a more blunt phenotype, and you have other mutations that occur also in regulatory regions that we are not considering. But in knockout, you have all the systems playing. So sometimes in that case, you have only one mutation, one gene leading a phenotype. I think that sometimes the phenotype in knockout mice would be milder, I don't know how to say this in English, than what we observed in EMU, that we have a more complex situation.
Okay. So last question. Again, on candidate 8, I noticed that in your early classification using this thing called candidate explorer, that parameter called this not good candidate. Why did you then take your time to knock out this gene? Like, what's the usefulness of the candidate explorer in guiding you ultimately?
Well, candidate explorer is a tool developed in molecular lab, and is a machine learning tool that consider, I talk a little bit in methods, but consider some points in some criteria of the phenotype in the pedigree, compare with the wild type, compare with the controls, but the final test is the results from knockout mice. And as for our screening, we do not have so many knockout mice. assessed, this tool is not very useful in our screening because we don't have enough data to train the machine learning tool to identify those candidates. It's because in molecular lab they have higher saturation, they develop a lot of local mice for immune system. And I think for immune systems, for the phenotypes that they assess that, I think an Explorer is a very interesting tool, but we do not have much knockout mice to train this tool. So I know that the majority of them show that it's not a good candidate, but I don't think we have enough information to use this candidate explorer.
Yeah, okay, good. So
it's I just consider the information here for tenacity.
So one quick last question. It's a picky one. So I noticed that the baseline systolic blood pressure between candidates, so like the average blood pressure in one strain compared to the other candidate is quite variable, or it is variable, at least 10 to 15 milliliters of mercury. So how do you take these into account when you analyze and interpret your results?
The variability of blood pressure between
categories. Yeah, if the average blood pressure in one strain is 130, in the and then in the other one is 115, they are starting from a very different level. So when you knock out those genes, you're going to compare now a change in blood pressure to very different baseline levels. And yet these mice are of the same genetic background, presumably. So it has to do with the variability of the trait presents naturally, right? The day of the week that you are doing the measurements, who knows what kind of parameters, but like everybody that work with blood pressure know that this is a quite variable phenotype. But now you're trying to make comparisons. So how does it go?
Well, usually we compare within the experiment. The only exception is the super pedigree analysis, that when we are considering different families and a whole family, the blood pressure is set higher than the other family, there is a normalization of those data. But usually we consider only the family. So I cannot compare directly two families. It is difficult. It is why when we consider different mutations in the same gene, in two or more families, we do this normalization to compare the results. But usually I do not compare data from different categories.
In the study design, did you cluster these animals in batches where the wild type and the knockout were measured at the same time for an individual candidate? Say candidate 8 wild type and candidate 8 knockout. Were the mice measured the blood pressure, their phenotype at the same time?
Yes, always. So for a family, for example, for a family EMU screening, we always phenotype together the family in the same time. So usually we do a family entirely, and when you finish a family, we take another family. And in knockout, I always use the 3 genotypes together. So sometimes I cannot do the experiments. For example, direct measurement, I cannot do all the experiments in the same day. So I usually use one homozygous Y type, heterozygous, and one homozygous mice from the same gender, because we assess both genders.
Yeah.
So we consider that.
That's great, Samantha. Thank you very much. I'm going to stop here.
Okay, thank you.
Good. Thank you, Marcelo. Interesting thoughts also about the problem. And now I will pass to Onushiki. Luis Onushiki is a professor here at the medicine department. His main interest is in nephrology, and he participated early on the time, also in the early 90s, that we're doing linkage analysis to pinpoint genes. He participated on an international effort while he was in the desert at Hopkins, and had opportunity to coordinate an effort to clone and characterize one of the genes associated with polycystic kidney disease. Then he came back to Brazil, and he continued on this, and he's clarifying or expanding the understanding of the pathway that leads to that defect. So he has also a good understanding of the not only the reductionist approach, but also to bring to the patient level. So I thank you again for participating on this, and now the word is yours, Luis.
Okay, so good afternoon, everyone. And I thank you, Peter, and I thank you, Samantha, for inviting me to be part of this committee of your PhD thesis defense. It's a pleasure to be here and to have the opportunity to follow your steps on this research and see how much you did since your qualifying exam. So congratulations to both of you. And I can see that...
Luis, may I just interrupt you? Just for people to know, Luis has been participating, always one member of the committee participates from the qualifying up to the completion of the work. So at this, in this event, Luis Onushiki is also the memory of the discussions that happened in the first, let's say, presentation of the data till now. So sorry about that.
Yeah, so that's it. And I can see how much work you've done and how robust data that you generated. And it's clear that you have generated data that are going to be used by you and many other people along the next years. So I like your text. It's very good, and I like very much your presentation. It was clear, it was well structured, and the data were appropriately presented. So having said that, I'm going to mention two things. First of all, I will not go over some comments that I had made previously on the qualifying exam. I'm going to just go over your points, and I'm going to try to avoid some points that I would raise, but have already been raised by Professor Beutler or myself. So, but in some issues I will touch again because I have to do that. Okay, so I'm going to bring just 2 points in methodology. Well, I see when you talk about the exon sequencing on page 19, you say here that you target total 60 megabases, including 50 base pairs of intronic sequence upstream and downstream. Okay, and that leads the next page to your sentence in the end of the first paragraph, stating that synonymous mutations and mutations not predicted to affect splicing or protein sense or also eliminated. Fine, sure. My point is that since you were already initiating this protocol, and given the nature of hypertension phenotype, including many genes, quantitative approach certainly applicable to many of these genes, and so why not using exon enriched kits to include like regulatory regions, such as, for example, there are some that come with promoter and UTRs, but at least promoters that are available and could be important for that type of phenotype, not only to be analyzed at this point, but to be integrated with your data along the time.
Well, first, yes, those variants are very important, and we are highlighting them, some of them using dual studies integrating with regulatory regions. But the main objective of this project is to identify genetic variants that cause a big effect size. And usually, those variants that affect regulatory regions does not show an effect size equal of the coding regions. It is what we identify in rat studies and human studies.
That's usually what happens, right?
Yes.
But for example, once you identify some, you might have with, if you had that approach, some additional data to be integrated.
Yes.
To later on, depending on what you could see, right? Isn't that true?
We can't integrate all the genetic variants, the synonymous variants that were defined in code regions. But as we do exome sequencing, we cannot identify genetic variants that occur in regulatory regions.
Yeah, but that's what I'm saying. If you had used this exome enriched kits, then you would have.
Include that.
Yeah.
Okay.
What I'm saying is that why not to use instead of the classical exome sequencing, this exome enriched, so that you would include along with your access regulatory regions such as say promoters. So you have also the promoters. I know that, so that it means that it's more expected to find these variants in the coding regions. I agree. But in some circumstances, you could find variants, like, for example, the promoter, that would be meaningful. And that could be not only be add more to your picture, but could be integrated with other data generated by others. It's just a matter of starting with, instead of an exome, with an exome enriched approach, including regulatory regions. Just clearly.
To include after the analysis.
Oh, you could include it before the analysis.
Yeah.
You could include it before the analysis, just so that you have that as well. And even being less likely, you could even find variants that could segregate with your phenotype.
Mm-hmm.
Right?
Yes.
So, do you guys ever consider that, or do you think that it's just to expand too much in that side? Was just, I think that you must have thought about it, but you said, well, for now, it's expanding a little bit too much, and let me stay here. Is that what happened?
Yeah, I think we focus in genetic variants that cause code.
If you're starting now, would you include it or not?
I don't know. I don't think so, because we are focusing this type of mutation. We focus to identify.
Better money.
Yeah.
Yeah.
I don't know.
Yeah. Because we already.
Right. Okay. Because you have to know. Okay. So it was an economical issue. Okay, but that's something that you would consider. The other thing that I would bring is that in your analysis, you mentioned here, for example, you can do it. So the approach of using with one of the tools, programs about applicable to predict pathogenicity.
Mm-hmm.
Okay. One thing that is important to know is that these programs have been developed to analyze variants in mendelian diseases. And then with the years, they were progressively improved to be applied also in polygenic diseases. But one thing that, of course, we have several tools here, but I think that we have to be aware of the limitations of this tool in a phenotype such as hypertension, polygenic, many genes, multifactorial, continuous variable.
Yeah.
There are several things that make it really and likely small effects. So it's something that you also consider that you may be working with a tool that is not ideal, helpful but not ideal, right? Okay. I'd like to come to go to page 43 and say that bringing the point again about the resistant hypertensive patients and all you said about it. Okay, you describe all the analysis, so and then you come to only 8 variants within 7 genes were considered like disease causing. And well, there are all this discussion you had with myself, fine. But I want to bring one point that's important. All mutations appear in heterozygosity. Okay, and this would match with, for example, the edited model, fine, right? And you did see that, but you see several, as you mentioned, cases for the variants or for the knockout models, like the phenotype, in one way or another, being associated with homozygosity, right? And not with heterozygosity. Okay, it's not a surprise to me that we found them all in heterozygosity. When we're talking about hypertension and the impact on the society and the population as a whole, right? Because this is much more likely to occur than to follow a recessive pattern.
Yes.
Okay. Okay, but then you see for most cases a phenotype that is significantly different for the homozygous, right? And not for the heterozygous. My question to you is that there is a reasonable possibility that these heterozygosity may be relevant. It's just that you were not able to catch it statistically within your pedigree.
Yeah.
Right? So then I bring the point of my colleagues previously. And one way of looking at that is to make combined animals. And you do have them. So, and catch them and combine 2, 3, 4, or 5 heterozygosities. And then you could show that they work. And even you can combine it in different ways to find out which of these heterozygotes would be meaningful for hypertension.
Yeah.
And you have the tools.
Yeah.
So I'm thinking about it.
Yeah.
Will you consider that?
No, I don't. I don't know if
But will you consider now?
Yes, of course. Yes, because I think what happened in human population is maybe they are presenting many genes with heterozygosity. Oh, I'm sorry. And maybe this is the way that that happens, the presentation of the phenotype in
Right.
population. And it is a very interesting point.
And not only you could reach significance, but also you could show maybe synergy or just an additive effect, and then you could come with some beautiful model.
Yes.
Integrating data that that could be achieved with tools that you already have.
Yes. Yeah. It is, as Dr. Bertzler said, to use those two genes, for example.
Yeah, it's just it's another angle of the same idea. Yes.
Yes. Nice.
Okay. The other thing, Samantha, because I mean, this is the type of pieces that I mean, you start coming with ideas. It opens too much. It opens too much. Your mind. But another thing, let me see here. Okay. I'll say. Okay, just before going to discussion, I'd like to hear about another thing. You guys certainly, one thing that we always think about is, what are you really looking at? Are you looking at systems that control blood pressure, or are you looking at hypertension? You are looking at both things. Right, because sometimes we pull it a little bit to the side, a little to the side, but at last, this is a physiological, pathogenic project, right?
Yes.
So when you do have for these hypertensive patients, their axons, right? So and you do have this complex design of the systems involved in the regulation of blood pressure out of sense, as you showed. Okay. Are you prepared or planning to go and looking at the things and having validated these genes, looking at regulatory genes of these, and looking at variants at such genes in these patients?
I don't know if I understand.
For example, you validated genes, right? They are related to blood pressure regulation, right? One way or another. Okay. Then you have their corresponding and already non-regulatory genes. And you have the exons of such patients.
Oh, yes.
Are you looking at variants in these regulatory genes in these patients?
Not yet, but we want to.
Because I do it
Yes.
because
we want to do. Yes, we want to see the directly genes involved in the pathway, the genes that regulate and each genes, those genes regulate or something like that, to see if you can find also genetic variants. Yes, of course. Yes, we want to do that.
Yeah, this is something also that you have that pulling hands and a potential to get very interesting things.
Yeah, we are starting with the candidates, but we are going to do that.
Right. Okay, yeah, great. Okay, another thing, this is more methodological, but it's important when we are looking for genes directly involved in hypertension is that we're looking at genes that are involved in primary hypertension, right? So one important thing that I think you mentioned is that when you get the animal phenotype, it's very, very important to have a comprehensive readout of the phenotypes like kidneys, heart, vessels, central nervous system, and adrenal, breathing. Why? Because you may just be facing a secondary hypertension.
Yes.
Such as, for example, in the homozygous for the MAC8.
Yes, exactly.
It's
It's visible.
okay. And another thing that is important not to be caught is to look at the corresponding heterozygous, because to make sure that if they do have an effect, it's not a smaller effect of the homozygous, but if it is, it's also going to be put out. But if it is not, then you're going to be following up on that. So I think that this type of approach does require a very careful phenotype not to be caught down the road.
Yes.
And you guys be doing that. One thing that you well discussed is that, I mean, of course, you guys went into the right road, which is the knockout approach for validation. But you discussed really well in your discussion that in some cases it's not the appropriate thing. For example, probably in some occasions, right? You go, for example, you require an overexpression point. Which profile of gene, for which profile of gene would you go for the overexpression of? Along the genes that you have. I mean, there are specific profiles that would make sense go for overexpression, right? For others, not. Which profile would you select overexpression?
Overexpression, maybe those that are more lethal, because with knockout mouse, we know that we cannot assess. I think overexpression are important, or time-dependent knockout mice that you knock out the mice only late on line and not during the development. Also, overexpression, maybe
would you agree that that would be an approach in some cases where you could not see a phenotype with the knockout approach?
yes.
Of course, you.
Yeah.
You'd have to.
Yeah.
Right.
I was imagining like
Yeah,
before the knockout mice reaching the.
No, but I'm saying that because that,
of course.
but of course you would have to be. It could not be obviously a truncated mutation. You would have to see whether that missense, for example, would have any possibility of just increasing activity,
Yeah.
or you'd have to, because you have to separate whether that would lead to increased activity or else if it was something that would lead to something completely different, like a gain of function, like with a gain of function associated with a network thing, it would also not make sense, right? So you have to see this specific event.
Yeah, one of the cases is one of the knockouts that we developed that is not tree. Let me see the name of this candidate. Candidate 27. Well, there is a disease, a related vascular disease that causes a mutation that is not a stop codon, but the protein remains in the membrane. So in this case, maybe it would be a good one to overexpress or even do a different mutation that recapitulates this phenotype. I don't know, but it's a very good strategy for this one.
All right. One thing that I was going to bring you to do, because we got a comment on that, but the issue about the susceptibility loci, right? Because we are always talking about a gene related to regulate up or down blood pressure. In this case, it's called blood pressure. Okay, but you may have, which is in the polygenic status, loci associated with susceptibility, right? And if you do a regular one, a screening with the approach you did, you make this, and this is particularly interesting because this is a multifactorial disease, and that when I talk with susceptibility, it can be applied to background, in that background or the environmental factor, right? So the point here is, with that, you've done, for example, in one word, just well done, like a check, different intake of sodium, fine, great. But the point is, for some susceptibility genes, you just get, if you get your design from scratch, from zero, for example, submitting all animals to household diet, right? These would not be caught, right? And I think that this is another issue that I can bring to you and say, well, this is also something that will be very helpful. And I think that you probably considered that, but it was also a matter of costs, because that would be an additional way of looking at the full picture of blood pressure regulation and hypertension. Correct?
Yes, correct. Well, the use of plethysmography put stress that it's not entirely based on conditional because of this stressful animal holders or animals are a little bit stressed, but exactly, it would be interesting if you can
do it there, so that it would catch them.
Different diets, but it's not possible.
Yeah. Okay, let me just
one thing that's important that from day one, Bruce was kind enough, because these animals, these pedigrees, goes to several protocols.
Yes.
And since we don't want to have any interference, we are the first one to phenotype.
All right.
So this is something that is quite important in the context of the way the thing goes. And the other thing is that even though the holder, I mean, if cost was not a problem or anything, we could think in doing all in telemetry and having real data. But I assume that we will not pick it up, the genes that we are picking up. Because when you have the animal with all homeostatic controllers in place and you do a small perturbation, perhaps we need the second hit. And the second hit here during the screening is really the stress. So it was fortuitous, but at the end of the day, I think if we would have no problem, no issue with money, whatever, we would not succeed in getting the genes that we get. Because, I mean, you would have a situation that would not favor that. Say, Bruce.
Yes, the question about extending the exome definition to include promoter elements, same story. We'd love to do it, but we can't afford it.
Okay, I see. All right, thank you. Well, I like the biology of this gene 27, you know. And I think that it's very possible that this gene could play a role as a susceptibility gene. Taking just because looking at it as a member of the Notch family. So one thing that will be probably interesting is to study the heads of my soul here. I can do that. I don't know whether I thought.
The sperm is in the way.
Yeah, we are bringing them.
So we are very anxious to. This is one of the animals that we want to play first.
Great. Yeah, it's beautiful. Yes.
There is this is one gene that we have to think twice if we're going to continue, because there is data, there are data in the literature. People are looking at especially on the vascular, in kidney function, and they are not. And there is one knockout that they didn't find changing blood pressure.
Yes.
So it's one gene that we are so curious about the biology that we are going to go for it. But I bet you that when we find the mechanism, people will say, "Oh, this is already expected." It's one of those things that eventually will be in trouble for us because it's so interesting. And there's some data related, but they're not going to the right target, I think. And because we have the data that really motivates us to go. But I bet you that when we get there, people are going to say, well, this is not so new.
Yeah, because there are some evidence.
Yeah. But I think from the biology point of view, it's very attractive.
Hopefully it's not going to be the reviewer.
Yeah, hopefully.
Okay. Well, and then I just to close my question before a last comment saying, I'm going to be making some comments, but about a topological approach to clarify regulatory systems, to clarify gene identification, on all of our discussion, and possibilities that, as I said, you were looking at these two things. But I think that I agree with Marcelo. I mean, you can also look at verbal issues because it allows you to do that. So I think that developing pharmacological approach with to reach these three aims would be a very nice track. And I'm not going to say more than that because Marcelo has addressed that really well. So, but just remembering that it can be really, it can really go to several tracks and really very nice things. Okay, so this opportunity was very good, not only to follow all of your work, which is beautiful, to have the chance to meet Professor Beutler and Marcelo. But I'd like to know, now on, right, what you're going to be doing, like as your next steps. I know from the research that that's clear several of things that we're going to be doing. But you have mentioned there the next steps and so, but not only these next steps that you went out in your presentation and discussed with us, but also how are you planning with reporting these things?
For articles.
For articles and so, yeah.
So this is the difficulty of this data because we have here a beautiful strategy, a lot of genes, but we don't want to publish all the genes to give the fish for everybody.
Right.
So I imagine to maybe write a perspective during the opportunity to do these analysis, and after that publish each knockout as individual paper showing the molecular mechanism in which they are involved with blood pressure regulation. And when we have more information about more genes identifying using this strategy together with some sites in human population, you can publish a more big article explaining better how we see the hypertensive phenotype and blood pressure regulation. This is my point of view.
So you were saying that you were planning to get some biological specific papers for each of these genes, which makes sense, or in some instances, depending on what you find in the human samples, go and strategize these reports related to the human findings, and that could be made without being so deep into the biology. So it's going to be depending on what you find with the approach that you mentioned.
Yes,
That is
yes.
okay.
But I think we can, there are some projects that we are doing here that we are trying to bring both things together, human studies and animal studies, and try to have some insight what's happening in the genetic architecture of blood pressure regulation and hypertension. But I think for this we need more information and more genes to have more insights about that. But I think we can focus in each gene and understand the mechanism that we have until now.
So thank you, Samantha. Thank you, Krieger.
Thank you.
It was very nice to be here again. Congratulations.
Thank you.
Well, now we go to the final phase, and I don't know how it's going to happen with the people that are in YouTube, because they are going to be.
No, I need to call the guy in the University of Medicina.
Yes.
And set to close the presentation.
The public session, and then it will be only us.
Yes. Just give me a minute because I need to call them.
Okay.
Close the section.
All right, perfect. So for deliberation, we have to just wait until they close the external part of the presentation, because I mean there are people watching this, and we are, the committee is going to just discuss to give the grade or to decide about that. So I will then, when this is done, excuse the people that are watching us, and we're going to be only us here. But yeah, I think several of the issues that you raised are quite important, and of course we are dealing with this. And Marcelo, I just want to get back to one point that you made when you were talking about you were asking Samantha about the target of statin and why that would not be predictable, right? At the populational level. I think you made one point, and the other one that's interesting, and perhaps a little bit different here, is that we are testing this under this
Yeah, so we have a foot of snow.
You were safe, right, Bruce?
It has been very mild otherwise, very mild wet winter.
It's down in Dallas, very nice. We had an inch of snow one day.
Here it's pretty hot right now.
Yeah.
And often you have like a storm at the end of the day or middle of the day.
Yeah, I like those. I remember those.
Yeah.
Like straight out of the Bible, like one of these storms. Like
That's right.
and
A couple of trees come down.
then it goes away. Yeah.
And then
Like
1
nothing.
hour later, everything's okay. Yeah. Okay. It's on already?
Yes, people said yes.
Okay, good. So thank you all for being here. And now I have a great pleasure to inform that committee evaluated your performance and suggested that you should receive your degree. So congratulations.
Thank you.
Good work, Samantha.
Thank you, Dr. Boyd.
And again, in the name of the graduate program of the medical school, I really want to say thank you for you, especially the ones that are far away, Marcelo and Dr. Bruce, and of course from here, from Luis Olushiki. And Samantha, we all hope that you're going to have a bright future. Marcelo wants to go where you're going to go next.
Yeah, I will stay here.
Yeah. Well, but I mean, it's about to do some. No, I was telling Marcelo by texting that, I mean, Samantha has already been hired, so it's not so easy for him to catch her up now. So,
Congratulations, Samantha.
Thank you.
good.
Thank you all, and I want to thank you everybody from this lab and Dr. Boyd's lab for all the development of the animals to help me with all the experiments and all this, the postdocs and PhD students and master students that are here, help me daily, and also to my boyfriend and my family to give me strength to continue in research and support. So thank you everybody, and I'm very happy.
Good, nice. Congratulations.
Thank you.
Yeah, and with that, especially for our friends from far away, thank you again, and let's have a great day.
Thank
Yes,
you.
same to you. See you again soon. Bye-bye.
Yeah,
Bye.
bye-bye.